Welcome everyone to this new session where we will discuss about item 1 of according to Bangladesh medical syllabus. Okay, so this is a read with me session where I will be reading or going through the book and you'll follow along. So let us begin with this chapter. That is chapter 2, structure of bacterial cells and its classification. So beginning with, what is bacteria? So far we have discussed that bacteria are unicellular, prokaryotic, free-living microorganisms. Now what is unicellular? That is, they just have one cell. What do you mean by prokaryotic? Pro means primitive and prokaryotic basically means primitive nucleus. Okay, primitive nucleus. And they are free-living, obviously. Are they parasites? No. What are parasites now and what are bacteria? Okay, firstly, parasites are the organisms which depend on host for shelter and nourishment. Okay, do bacteria depend on us humans? Are they always in us? No, they are free living. They are found wildly in our world. Okay, so they are free living microorganisms. They are not parasites. They are unicellular and they have one nucleus. That is like they have primitive nucleus. Okay, going into that primitive nucleus thing, I have already explained million times that a bacteria has cell wall, it has cell membrane, and it has some chromatin material, and it has ribosomes. This is a basic structure. Now, normally what do we see in us or in eukaryotic cells? There is one nucleus which has nucleoli and chromatin, right? Nucleoli and chromatin. But here in case of bacterial cells, do you see any cell membrane? Any nuclear membrane? No, there is no nuclear membrane. So any structure without this nuclear membrane is basically a nucleoid. So nucleoid is any structure having chromatin material, but it is not bound by a membrane. So it is not a nucleus. It is not a true nucleus. It is basically a collection of chromatin. And that collection of chromatin, this amorphous collection of chromatin, is known as nucleoid. Okay, is known as nucleoid. And in case of eukaryotes, this is nucleus. Got it? So what do bacteria have? Bacteria have nucleoid. And what do humans have? Or what do fungi have? They have nucleus. Got the basic difference? So they have primitive nucleus, that is nucleoid. And other structures that you find is ribosome, which is very important, and cell membrane and cell wall. These are the 4 essential structures that you will find. Okay. So bacteria are primitive, they are unicellular, they are free-living organisms, and they contain genetic information and energy-producing and biosynthetic system necessary for growth and reproduction. Because they do not depend on us, they have their own biosynthetic system for energy production. Okay, and they divide or reproduce by simple binary fission. Before starting this session, I would like to request you all to watch my previous videos on general bacteriology. After watching those 2 videos, your concepts will be clear, and after watching those videos, you can follow up with this session. Thank you. Okay, so bacteria's like basic definition is done. What is important is the classification. Okay, now some bacterias you'll find round in shape. Those are known as cocci. Cocci means round. Then you'll find some rods. Okay, those rods are called as bacilli. Then you may also find something in between, which is cocobacilli. Clear? Coco means round, bacilli means rod, and cocobacilli is in between. It is in between rods and bacilli is cocobacilli. Okay, now let us see the organisms. Okay, so if this round cocci are arranged in clusters, okay, this is like grape, right? How do you find grape? Grapes are in clusters, right? Like this. So anything that you find in cluster, that becomes Staphylococcus. From now on, I want you all to memorize this name. Staphylococcus is found in grape-like clusters. Next, if it is found in chains like this, okay, you know how chains are like. If it is found in chains, various forms but chain form. Okay, this is known as streptococcus. Okay, staphylococcus and streptococcus are different things. Staphylo means grape like, strepto means linear. Okay, now why are some staphylo and why are some strepto? You can see I have already said that bacteria they multiply by binary fission, right? 1 becomes 2, 2 becomes 4, and 4 becomes 8. Likewise, so if these bacteria, for example, if this bacteria has to multiply, if it multiplies in every axis, okay, then it forms clusters, right? But if one bacteria just multiplies in one axis, then it forms chains. You got the point? If one bacteria multiplies in every possible axis, okay, then it will form lots of clusters. That is, it will form cells all around it. But if one bacteria just multiplies in one axis, then it will give rise to linear structure. Okay, so because of multi-axial growth, staphylococcus are in clusters, and because of uniaxial growth, streptococcus are in chain. So uniaxial growth and multi-axial growth. Okay, these are the basic differences. Now, you are familiar with staphylococcus and streptococcus. Then, streptococcus only. Okay, they are linear. I said that already. But if only two are found, okay, then we call it diplococci. That is two cocci. Two cocci. Okay, so cocci are round in shape. They can be two in number, or in chain, or in grape-like clusters. Grape-like clusters, that is staphylococcus. Chain is streptococcus, and the ones who remain in pair, okay, are streptococcus pneumoniae and Neisseria species. Now, going to streptococcus pneumoniae and Neisseria species, what I mean to say is, okay, what I mean to say is, I said already that if you find it in chain, that is streptococcus. Okay, streptococcus, it is one species, it has lots of organisms. Okay, streptococcus is found in chain, but one of the streptococcus, okay, streptococcus pneumoniae, it is found in pair. Okay, it is found in pair. Clear? Streptococcus pneumoniae, it is found in pair, that is two cocci like this. So these are known as diplococci. Clear? Now, rods, talking about cocci. Okay, these cocci, if you have watched my video on staining, you will be familiar with what is gram stain positive and what is gram stain negative. Gram stain positive are the organisms which are purple against red background, and negative are the organisms which are red against red background. You know this so far, right? So purple against red background are gram positive, and red against red background are gram negative. So if anyone asks you, name some gram positive organisms. Gram positive organisms, invariably you can say gram positive, if they make it specific, gram positive cocci or gram negative cocci. If it is gram positive cocci, then you can say staphylococcus and streptococcus. Okay, staphylo and strepto. Remember it right now. Staphylo and strepto are gram positive cocci. Then what is gram negative? Gram negative is Neisseria. Neisseria species. Okay. Then what is this Neisseria species now? We don't know anything about it. About strepto, we know that they are linear. Staphylo, we know that they are in clusters. We know why they are in clusters and why they are in linear arrangement. And about streptococcus, we also know a special type, streptococcus pneumoniae, which is in pair. Clear? Then what about this Neisseria? What is this now? All we know is that it is gram negative. Gram negative, that is, it is red against red background. So now let us know more about Neisseria species. Neisseria species, as the name suggests, it should be nice, right? Neisseria species, It should be nice, but it is not nice. Not nice in the sense that it is of two types. One is Neisseria gonorrhoeae, and another is Neisseria meningitidis. Okay, two species. Mainly, there are lots of species, but we'll just focus on Neisseria gonorrhoeae and Neisseria meningitidis, which are the two types of Neisseria. What are Neisseria? They are gram-negative organisms, gram-negative cocci. Neisseria has two types, gonorrhoeae and meningitidis. The basic difference is, they are both diplococci, okay? They are both diplococci, and they are found intracellularly, where inside the monocytes, okay? So, or macrophages, you may say, they are found intracellularly, okay? And they are diplococci. 2 points, intracellular and diplococci. So, what is the difference? Like, how will we differentiate gonococci and meningitidis based on its shape? I said they are diplococci. How will we differentiate? The basic difference is meningitidis is lancet shaped, okay, and gonococci is kidney shaped, okay. One is kidney shaped, the other one is lens shaped. Clear? It looks like your sunglass, right? Lens shaped. You got the difference? G, kidney shaped, and the other one is lens shaped. Okay, this gonorrhea, it causes infection in the genital area, so it is found in genital region, okay. And meningitidis in the brain, it causes meningitis in brain. So yesterday also I mentioned, any organism that causes any infection in brain, it will be capsulated. So Neisseria meningitidis is capsulated, but this gonorrhea is not capsulated, okay. Basic difference between Neisseria are of two types mainly, gonorrhea and meningitidis. Gonorrhea, it causes urethritis, cervicitis, or the infections in the genital tract. They are kidney shaped. And meningitidis, they cause meningitis, therefore they are capsulated and they are lens shaped. But mind you, both of them are diplococci. Okay. So diplococci, I hope I made it clear. So morphological classification. Morphology, what do you mean by morphology? It basically means shape. Okay. So morphology means shape or anatomically. So morphologically, cocci can be diplococci found in pair, which is Streptococcus pneumoniae, Neisseria species, both gonorrhea and meningitidis, both are diplococci. Then in chain, Streptococcus species, in cluster, Staphylococcus species. Okay. Then what are bacilli? Bacilli means rod. Very good example, acid fast bacilli, that is Mycobacterium tuberculosis, or you can also go along with Enterobacteriaceae. Okay. Then spiral and pleomorphic. Okay, spiral, some bacilli can be spiraled and some can be pleomorphic. Pleomorphic means that they are variable in shape. Okay, one very good example of pleomorphic is P for Proteus. P for Proteus. Okay. Now let's move forward. On the basis of staining, they can be gram positive. Gram positive are violet color on gram stain. That is, they are purple against red background, and negative is red against red background. I mentioned already, gram positive are Streptococcus and Staphylococcus, and gram negative are Neisseria species. Done. Please watch a video on staining beforehand. Okay. Then on the basis of spore formation, spore forming can be bacil. Now, okay, what is spore? Firstly, if this is any bacteria, when it knows that the oxygen or nutrients are getting depleted, when its death is approaching near, then what it does is it forms resistant structures. Okay, resistant structures through which it will survive, and later this resistant structures will form the vegetative form, that is the actual form, and they can produce their progeny and survive later on. But the spore is one resistant dormant form. It does not multiply, it does not do anything. One resistant dormant structure formed by bacteria is known as spores. Do all bacteria form spores? No. So mainly Bacillus BC. Okay, Bacillus and Clostridium species, they form spores. Bacillus and Clostridium, they form spores. Okay, Bacillus and Clostridium, they form spores. Then non-spore formings, that is, they do not form spores. Strepto and Staphylo. You know about Streptococcus. Did I say they form spores? No. You know about Staphylococcus. They also don't form spores. So you now know the example of spore forming and non-spore forming. More into the spore forming, Bacillus species, okay, they form central spore. Okay, bacteria, they always form endospore. Endo means the spore is inside. Okay, ecto means the spore is outside. Bacteria form endospore, and this endospore is centrally aligned in case of Bacillus. Okay, central spore is found in case of Bacillus. Clear? Then Clostridium. Clostridium is lots of species in it. Okay, you are familiar with tetany, are you not? Tetany is the one which causes tetanus. Okay, there is something else also, perfringes or tertium or histolyticum. Okay, just be familiar with these names. So these tetany and all, T for tetany, T for tertium. So these have terminal spores. Okay, terminal spores. Terminal for T for terminal, T for tertium. And depending on the shape of the spore, if it is round or oval, the shape will change. Okay, so if it is round, it will be like this. If it is oval, it will be like this, right? So depending on the shape and structure, they can either be, depending on the spore, okay, depending on the spore, they can either be tennis racket form, or they can be drumstick form, right? So they look like drumstick, they look like tennis racket. So if the spore is round or oval, depending on that, they can be drumstick form or tennis racket form. They may look like that. Okay, they are not actually drumstick or tennis, but then like in the microscope they look like, they give the appearance of drumstick or tennis ball because of the tertiary, because of the terminal spore. Okay, for now all you need to remember is that there are two species of bacteria which produce spores. One is Bacillus, the other one is Clostridium. If you can remember, just remember Bacillus has central spores, and another thing, spores are always endospores in case of bacteria. So are there ectospores? Yes, ectospores are there, but ectospores are there in fungus. Okay, and what is the use of spore? Spore is for survival, and in case of fungus, spore is for sexual reproduction. Okay, the basic difference. Now moving forward, so you classified bacteria on the basis of spore forming and non-spore forming, on the basis of position of living, if they are found extracellularly, or if they are obligate intracellular parasites, obligate intracellular bacteria. Okay, extracellular, that is, they are found mostly outside the cell, they live outside the cell. Okay, most bacteria do that. Only selective bacteria are the ones who live within or inside the cell. Those organisms which survive inside our cells are Rickettsia and Chlamydia. Rickettsia and Chlamydia. Now, on the basis of cell wall, all the bacteria have cell wall except Mycoplasma. What does Mycoplasma have? Mycoplasma have sterols. Okay, it does not know cell wall. So this classification is also done on the basis of cell wall. Now moving forward, this is the structure of a typical bacterium. Let me draw it again for you. This is cell wall, this is cell membrane, this is the nucleoid, and this is ribosome. Okay, now ribosome, as you all know, is 70s in case of prokaryotes, and in case of eukaryotes, that is true nucleus organisms like fungus and humans, it is 80s, but in case of bacteria it is 70s. This is the chromatin, which is circular, as you can see. This is mesosome. Okay, and this is cell membrane, this is cell wall, and additional structures like villi can be present, and flagella can also be present. You know the basic structure. So now this is the chromatin here. Okay, this is the chromatin here, chromosome. These small things are the ribosomes. This hair-like protrusion is the pilus. There can be inclusion bodies. This long structure is the flagella. Okay, plasmids are extracellular chromosomes which are present. Okay, cell membrane is there and cell wall is also there. Okay, and some bacteria are capsulated. Talking about the essential and non-essential structures in case of positive and in case of negative, this is a quick recapitulation. So this is the cell membrane, okay, that I'm drawing. This in case of gram negative is the outer membrane. Okay, and okay, this can be the peptidoglycan layer, and this is the peptidoglycan in negative. Okay, both can have and this is teichoic acid. Clear? And this space is the periplasmic space. Periplasmic space. Clear? This is the basic structure. Now we'll discuss more about it. This cell membrane or cytoplasmic membrane, you know, this peptidoglycan layer, yesterday I told you it is sugar based, and the arrangement is NAM-NAG, right? And it has side chain, and these side chains, like it has side chains, and these side chains are cross-linked with another NAM side unit, right? There are cross-links present. Okay, this is the side chain and cross-links are present, and this cross-link is the site of action of many beta-lactam drugs like penicillin and cephalosporin. And this NAM-NAG connection is the site of action of lysozymes. You know that, right? This is about the peptidoglycan layer. Just a quick recap. And outer membrane is present in case of gram negative, and it has two components. Okay, mainly lipid A. Okay, which is responsible for septic shock, and it has polysaccharides. Okay, now let us continue our discussion. So till this structure of the cell we have completed. Now let's go to the components. Cell wall is made up of peptidoglycan, which is sugar backbone, gives rigid support obviously, and protects against osmotic pressure. And important to note here is it is the site of action of penicillin, cephalosporin, and it is degraded by lysosomes. Penicillin and cephalosporin, as you know or recall, they act on transpeptidase enzyme. And lysosomes, okay, they are basically found in saliva, and they act on beta 1,4 glycosidic linkage, which connects NAM and NAG subunits. Okay, and transpeptidase enzyme is responsible for the formation of transpeptide or cross-link formation between two side chains of NAM. Okay, these are side chains, and this is the cross-link. Transpeptidase enzyme is responsible for the formation of transpeptide linkage or cross-link between the NAM-NAG side chains. Okay, clear? Then outer membrane is present in gram-negative bacteria, and teichoic acid is present in gram-positive bacteria. Outer membranes, this lipid A is endotoxin. It is responsible for shock. Okay, and it is polysaccharide or sugar. In all cases they act as surface antigen. Polysaccharides, if you ever hear the name polysaccharide, remember they will be used as surface antigen in laboratory for diagnosis. Okay, and teichoic acid, it is important for attachment, number one. It can also produce shock. Okay, in gram negative, lipid A is responsible for shock, and in gram positive, teichoic acid is responsible for septic shock. Teichoic acid is also a surface antigen used in lab for diagnosis, but it is rarely used. Important to note here is it is responsible for attachment and septic shock. Clear? Then cytoplasmic membrane, you know it is bilayer. membrane is always there for active transport of molecules and secretion of enzymes and toxins. Ribosomes. It is 70S in case of bacteria and 80S in case of eukaryotes. 70S, if we break it down, it will be 50S and 30S subunit. They are important for protein synthesis. Okay, and this is the site of antibiotic that act by inhibiting protein synthesis. You remember protein synthesis inhibitors that act in 30S and 50S subunit. Okay, 30S, the mnemonic was AT, which stands for aminoglycoside and tetracycline, and 50S includes chloramphenicol, clindamycin, linezolid, and macrolides. Okay, these are 50S proteins and these are 30S. Only aminoglycoside out of them is bactericidal, rest are bacteriostatic. Clear? Okay. This is basically a summarization. Okay, then nucleoid. Nucleoid is the genetic material. Why nucleoid and why not nucleus? Because there is no nuclear membrane. Next is mesosome, which is very important. Mesosome is invagination of the plasma membrane. You know that when I make this invagination, this is mesosome. It participates in cell division and secretion. Okay. Central mesosome is responsible for cell division, and other lateral mesosomes are responsible for secretion. Now periplasm, you know, the space between the inner and outer membrane is known as periplasm. It is important because it contains many enzymes that will destroy the bacteria, that will destroy the bactericidal drugs. Okay, so it has enzymes like hydrolytic enzymes, for example beta lactamase. Beta lactamase is present there, and this beta lactamase is responsible for degrading or damaging that beta lactam drugs. So beta lactam drugs like penicillin, cephalosporin are degraded by beta lactamase enzyme, which is present in periplasm, and periplasm is present in gram negative. That is why penicillin and cephalosporins, basically penicillins, they do not work in gram negative organisms. They are given in gram positive only because gram negative have beta lactamase in their periplasm, which can degrade beta lactam drugs. Clear? Okay. Then talking about the non-essential components. Okay, non-essential components include capsule. Capsule, all the structure, all the organisms going to head and to the chest. Okay, for now remember chest organisms: Haemophilus influenzae, Streptococcus pneumoniae, Streptococcus pyogenes, and in brain you can remember Neisseria meningitidis. Clear? They protect against phagocytosis. How? Because they form a well formed layer outside, right? So the phagocytes or the macrophages, neutrophils, they cannot go and phagocytose. Clear? Since they have a capsule, they are very important in lab diagnosis, and the reaction for their diagnosis is quelling reaction. Now what is this quelling reaction? Quelling reaction is nothing but if they have a capsule, remember this capsule is antigenic. That is, it is an antigen, right? It is an antigen. Now if we give any antibody, will it bind to this antigen? Yes. So when it binds, when the antibody given in lab binds with the antigen, that is the capsule, this capsule swells up. That is, it becomes bigger in size, and this bigger in shape can be observed in laboratory, and this swelling is known as Quelling reaction. If it has capsule, it will swell. If the organism does not have capsule, no matter how many antibodies we give, will it swell? No, it won't. So Quelling reaction is there to identify all the capsulated organisms. Clear? Okay. Then pilus or fimbria. What is pilus? Pilus, you know, are hair-like protrusions. They are glycoprotein. Glycoprotein, that is, they are made up of sugar and protein. Pilus, there can be two types: ordinary pilus and sex pilus. Ordinary pilus is just for attachment. Okay. One example is Neisseria gonorrhoeae. You are familiar with gonorrhea now, which is kidney shaped, and I told you it causes genital infections. How does it cause? It has pili, which mediates its attachment to the urinary tract. Okay, and it causes infection there. Okay, so pili is responsible for gonorrhea's infection. Clear? Then sex pilus. Sex pilus is there for attachment between two bacteria during conjugation. What is conjugation? Conjugation is transfer of genetic material. So imagine, if this is one bacteria, if this is another bacteria, through this fimbriae it is sending or transmitting information, and this process is known as conjugation. Okay, clear? This is conjugation. So there is sex pilus and ordinary pilus. Two type of pili are present, sex and ordinary pili. Sex pili is for attachment and conjugation. Conjugation is basically transfer of genetic material. And ordinary pilus is for attachment. Example, Neisseria gonorrhoeae, which is responsible for urinary tract infections. And this pili is the factor or the thing which helps the bacteria to attach to the cell surface and cause infection. Okay, then flagellum. Flagellum is for motility. Then spore. Spore is like thick keratin coat. It has like you know what spore is, right? Resistant structure, which is for resistance against dehydration, heat and chemical. Then what are plasmids? Plasmids are extra chromosomal DNA which contain gene for antibiotic resistance and toxin. So they synthesize or they form, they even have genes for pili synthesis or the synthesis of pilus. Then they also form antigenic antibody resistant genes. Okay, then granules can be present and glycocalyx. What is glycocalyx? Glycocalyx is polysaccharide. The basic difference between glycocalyx and the capsule is that capsule has a proper, it is properly membrane bound, right? Capsule is a proper structure. Glycocalyx is like irregular amorphous density, and glycocalyx is basically sticky. It helps to adhere to the surface. Okay, example, because of glycocalyx, in prosthetic heart valves, this Staphylococcus epidermidis adheres to the heart valves because of the glycocalyx. Okay, we'll come into it. Then what is nuclear envelope? Nuclear envelope are all these. Okay, look at this piece. This is the outer membrane. This is the inner membrane, you know, right, in case of gram negative, and this is the peptidoglycan layer, and this is the periplasmic space. Everything outside this everything outside this plasma membrane or the inner membrane is the envelope. So in case of gram negative, this envelope will consist of cell wall. Okay, look at the components of envelope of gram negative bacteria. Okay, gram negative bacteria is surrounded by inner membrane. Then it is surrounded by cell wall, which is composed of peptidoglycan layer, periplasmic space and outer membrane, and capsule in some bacteria. What it basically means is that if this is bacteria, it has cell membrane, then it has outer membrane, periplasmic space and peptidoglycan layer. This outer membrane, periplasmic space and this peptidoglycan layer, these three things are cell wall. This is cell membrane, and it can also have capsule. These are cell envelope components. Okay, cytoplasmic membrane, cell wall and capsule. And in case of gram positive, you know, there is cytoplasmic membrane and cell wall is composed just of peptidoglycan layer. There is no outer membrane, and capsule can be present in some bacteria. Clear? You know the functions. They are for protection. Okay, and responsible for metabolic processes, you know, for transfer, for attachment. Okay, cell wall, you know what cell wall is. It is made up of peptidoglycan layer, teichoic acid in case of gram positive. Then what is cytoplasmic membrane? Cell membrane, you know, cell membrane is bilayer. Okay, lipid bilayer. It is semi-permeable membrane, and cell membrane, like every bacteria has cell membrane, and they have cell wall also, but only mycoplasma has just cell membrane. Clear? Cell membrane of prokaryotes are distinguished from eukaryotes by the absence of sterols. Okay, so all the bacteria lack sterol. They do not have sterols. Okay, exception is mycoplasma. So what I mean to say is all the bacteria have cell wall except mycoplasma. Okay, mycoplasma is an exception. So since it does not have cell wall, it has sterol so as to compensate. Okay, but in case of eukaryotes, that is humans or fungus, we always have sterols. Humans have cholesterol, and fungi, they have ergosterol, but we have sterols. Bacteria don't have sterols. Only exception is mycoplasma, which has sterols. Clear? But bacteria, they don't have sterols, but they can have sterol like molecules, which are known as hopanoids. Not very important to note, but just know that bacteria don't have sterols. They can have sterol like molecules like hopanoids. They do not have sterols. Exception is mycoplasma, which has sterols. Because it does not have cell wall, it needs sterol for its protection. Function of cell membrane. Cell permeable membrane allows transport of substances. You know what cell wall is now. Cell wall is made up of peptidoglycan, teichoic acid, polysaccharides in case of gram positive, and in case of gram negative. Now note it down. It is made up of outer membrane, right? And now firstly, let me clear one concept. Okay, if this is cell, everything that is present outside is the envelope. Okay, envelope minus the cytoplasmic membrane is the cell wall. Clear? So everything outside, including the cell membrane, the real inner membrane or cytoplasmic membrane, is the envelope. But envelope minus the membrane is the cell wall. Clear? So cell wall will have outer membrane, right? It will have peptidoglycan. It will have periplasmic space. So yes, outer membrane is there. Peptidoglycan layer is there. Periplasmic space is also there. Then what is this lipoprotein and lipopolysaccharide? LPS are nothing but the molecules present in the outer membrane itself. Okay, they are nothing, but mainly we just have three structures in the cell wall. They are outer membrane, peptidoglycan, and periplasmic space. This lipopolysaccharide is nothing but molecules present in the outer membrane, so they are a part of outer membrane. Clear? And lipoproteins are basically abundant proteins which are found in gram-negative bacteria, which are joined to the underlying peptidoglycan and embedded in the outer membrane by its hydrophobic end, stabilizes the outer membrane of gram-negative cell wall. What it means is, if this is the outer membrane, there will be some lipoproteins which form covalent bond with the peptidoglycan layer and provide stability. That is, imagine like this. If this is outer membrane, it has some lipoproteins. Okay, these lipoproteins form bond with this peptidoglycan and are responsible for their stability. Otherwise, this peptidoglycan will float away, right? So for the stability of outer membrane and the peptidoglycan, there needs to be some bond formation which is formed by lipoproteins. Clear? So mainly three things are found: outer membrane, inner membrane, outer membrane, periplasmic space, and peptidoglycan in between that periplasmic space. Lipopolysaccharides are nothing but a part of outer membrane, and lipoproteins are structures which form the connection between outer membrane and peptidoglycan layer and help in providing stability. Functions of cell wall, you know that. Now, gram positive and gram negative cell wall. You know, gram positive is very thick, gram negative is thin. Gram positive is very thick because of peptidoglycan layers. It is 40 layers thick, and gram negative only 1 to 2 layer thick. Since gram negative does not have peptidoglycan layer, it is compensated by lipid, which is more in gram negative. Then teichoic acid is present in gram positive, and lipopolysaccharide, outer membrane, periplasmic space are present in gram negative. You know this so far. Now, if we treat with lysozyme, what happens? Gram positive will turn into protoplast, a structure, and gram negative will get converted into spheroblast. Clear? Now, what is endotoxin and what is exotoxin? I'm coming to that. So, bacteria, if they secrete any toxin, okay, they secrete. This is known as exotoxin. Exo means outside, though it means that they secrete the toxin outside. This is exotoxin. All the bacteria, positive or negative, they secrete exotoxin. But in case of gram negative organisms, you know there is outer membrane, right? This outer membrane, I said, has lipid A, which is very toxic. This lipid A itself is known as endotoxin. because it is inside the structure of the bacteria. Okay, so if it is inside the bacteria, if it is inside the structure of the bacteria, how will we get it? Will we get it by simply like secretion? No, it cannot be secreted. So only when the bacteria dies, only after the lysis of the bacteria, this endotoxin comes out in the surrounding and causes septic shock. Okay, septic means organism related shock. Okay, so lipid A in the outer membrane is toxic. It is basically endotoxin, which is responsible for septic shock, and we get it only after the lysis of bacteria, and it is found only in gram negative organisms. So endotoxin is only in gram negative organisms. Clear? So what are the enzymes that attack the cell wall? You know, lysozymes found in saliva, which attacks the beta 1,4 linkage. Beta 1,4 linkage. And autolysins are basically substances which are responsible for cell growth and division. And antibiotics like penicillin, cephalosporin, they activate the autolysins at all times and causes the lysis of cell wall, okay, and bacteria. Cell wall of acid fast bacteria, you know, they are rich in mycolic acids. Then peptidoglycans, I've already discussed this structure, so I think you'll be able to understand. Sugar backbone, NAM and NAG, N-acetylmuramic acid and acetylglucosamine, connected by beta 1,4 linkage. Side chain, which is tetrapeptide, 4 peptides. You can see 1, 2, 3, 4, 1, 2, 3, 4 peptides, okay? And these peptides are cross-linked. You know, there is cross-link. There are tetrapeptide side chain are cross-linked by a peptide cross-link, okay? You can see 1, 2, 3, 4, 5 peptide cross-link. Peptide cross-link. Okay, and it is done by transpeptidase enzyme, which is inhibited by penicillin cephalosporin. Okay, transpeptidase enzyme. And in case of gram negative, I said there is direct connection. There is no need of transpeptide crosslink. Why so? Because this in case of gram positive, this C is lysine, which is replaced by diaminopamelic acid in case of gram negative. So DAP can form direct connection with the other part. Okay, so full form of A is alanine. You can see here, right? A stands for alanine, B stands for glutamic acid. Okay, sorry, A stands for alanine, B stands for glycine. It should be written somewhere here. B stands for glycine, glutamic acid. Sorry, A stands for alanine, B stands for glutamic acid, C stands for lysine in case of gram positive, and it stands for DAP, diaminopamelic acid, in case of gram negative, and lastly it has alanine again. Okay, alanine. So to sum up, it is made up of AGLA, alanine, glutamic acid, lysine, and alanine again. And two AGLA, one lysine is connected to alanine and another by tetrapeptide side chain. Pentapeptide side chain. 1, 2, 3, 4, 5. Pentapeptide side chain. Okay. But in case of gram negative, there is direct connection. That is AGLA. This L is replaced by DAP. So there is direct connection in case of gram negative, and in case of gram positive, it is connected by pentapeptide. Okay. Now, what is lipopolysaccharide? You know, LPS. It is a component of outer membrane. So outer membrane basically looks like this. It has one central core polysaccharides. Okay. Then outside also it has polysaccharide. Inside it has polysaccharide. Outside also it has polysaccharide. Outside polysaccharide is antigenic. It is outside, right? So it is antigenic. It can trigger the formation of antibodies. It can bind with antibodies, and in lab we can identify it. And inside it has fatty acid. Okay. This fatty acid plus polysaccharide together is known as lipid A. And lipid A is responsible for the shock. Okay. So this diagram shall be clear to you now. If this is fatty acid, this is the core polysaccharides. Together we form lipid A. And outside also there is one polysaccharide which is known as somatic or O antigen. Clear? So lipid A and O antigen or polysaccharide together make the lipopolysaccharide, which forms the outer membrane. So outer membrane is formed by LPS. So functions: lipid A produces toxic shock, and somatic or O antigen is for laboratory diagnosis. Now, what is periplasmic space? The space between outer and inner membrane, present only in gram-negative bacteria. Space between outer and inner membrane, present only in gram-negative bacteria. And it is important because it has substrates. It helps in transport of specific substrates. It has hydrolytic enzymes like beta-lactamase, very important, beta-lactamase, which inactivates certain antibiotics like penicillin. Clear? Then what is teichoic acid? Teichoic acid is found only in gram-positive organisms. They induce septic shock and they are used as antigen. Mainly they are important for attachment. Okay. Now what is glycocalyx? Also known as slime layer. They are amorphous, viscid, colloidal coating. They are basically coating. They are not a fixed, structure. They are coating. If this is bacteria, this is glycocalyx. Glycocalyx covers the surface like a film. It is very sticky. Mind you, it is very sticky. It adheres firmly to various structures like skin, heart valves, catheters. So it mediates the adherence of bacteria such as Streptococcus mutans to the surface of teeth. Okay, so in our teeth, Streptococcus, that is Streptococcus by name you understand it is linear. Streptococcus mutans is a type of Streptococcus species. It can adhere to our teeth. Okay, it can adhere to our teeth because of glycocalyx. Example, Streptococcus mutans. Another, glycocalyx layer plays an important role in resistance. Glycocalyx producing Pseudomonas aeruginosa, okay, causes respiratory tract infections in cystic fibrosis patient. For now, just remember Streptococcus mutans found in teeth, and another Staphylococcus epidermidis, I've been saying this repeatedly, is responsible for endocarditis or affecting the heart valve. So you take two examples, Staphylococcus epidermidis and Streptococcus mutans. Staph, is found in clusters. Streps, found in chain. Streptococcus mutans found in teeth, and Staphylococcus epidermidis is responsible for endocarditis. Next, capsule. Capsule is a gelatinous layer. Gelatin is sticky, covering the entire bacteria, and capsule is always made up of polysaccharide. And one exception to this is anthrax bacillus. That is, anthrax bacillus is an example of exception. Okay, all the capsulated organisms are made up of sugar. They are made up of polysaccharide. Only exception is bacillus, which is made up of peptide or protein. Okay, D-glutamic acid. So just remember, capsule is a gelatinous layer. It covers the entire bacteria. It is made up of sugar. Okay, that is polysaccharide, and only exception is Bacillus anthracis. What is Bacillus anthrax? Now, Bacillus means rod, you know that. And I said Bacillus has spore, central spore. You know two things, and anthrax. So Bacillus anthracis is an organism which has spore, and it also has one capsule. And exception is that, funny thing is that it is made up of peptide. Normally all the capsules are sugary in nature, right? But this is made up of peptide, that is protein. Okay, so composition of capsule, you know that capsule is mostly made up of polysaccharide, often referred to as exopolysaccharides. Exo means it is found outside, right? It is found outside the bacteria. It is coating the bacteria. That is why it is exopolysaccharide. And they are neutral homopolysaccharides. Sometimes Bacillus anthracis has a capsule comprising of polyamino acids, okay, which is an exception. Okay, now capsule, it protects against phagocytosis. It protects against immune reaction. How? Phagocytosis, because it has capsule, nobody can go and eat it up. Okay, because it is in capsule, antigen is hidden. Okay, so when antigen is hidden, like core antigen is hidden, so antibodies are not formed. Less antibodies are formed. So immune response, it protects against immune response, that is, it protects against the development of antibodies, and protects against phagocytosis, that is, it protects against being eaten up. Then it provides adhesion to the host cell surface. Capsule is very sticky, helps in adherence. Then capsule itself is antigenic, I said that earlier, by Quellung reaction. If we give antibodies to this antigen, that is capsule, it will swell up and Quellung reaction will be positive. And this capsule is used in vaccines. Okay, example, pneumococcal vaccines. Various vaccines use this capsule as antigen. Okay, and are available in the market. Okay, serological detection, Quellung reaction. Okay, and various negative staining. Yesterday when I talked about staining, I said there is one positive staining, one negative staining. Negative staining is that in positive staining the organism will get stained. In negative staining, everything except that organism will get stained, and the organism won't get stained, right? So it is done using Indian ink, negative staining. And this is the example. This is pneumococci. Why pneumococci? Streptococcus pneumoniae, as I mentioned earlier, it is diplococci, right? It is diplococci, and it is capsulated. Why capsulated? Because it goes to lungs. It is capsulated. So when we give negative ink, because of this thick capsule, this bacteria won't get stained, but everything else will get stained. So you get a structure like this. This is the capsule. This white thing is capsule. This black thing is the bacteria, but because of this thick capsule, everything else will get stained, but the capsule won't get stained. So we can identify the organism, and this method is Indian ink or negative staining method, from which we identified pneumococci. Pneumococci, you know, is diplococci. Pneumococci is nothing but Streptococcus pneumoniae. Clear? And what is it responsible for? Pneumonia. Easy. Now what is Quellung reaction? You see the capsule here and you see the capsule here. Is the capsule swollen up? Yes. Why? Because when we give antibody, it got bound to the antigen, and the antigen antibody, it got swollen up. Okay, the capsule got swollen up because of the reaction. And this reaction is very important in identification of capsule or capsulated organisms. Capsules are antigenic, so in the presence of homologous antibody, the capsule will swell up greatly, and this swelling phenomenon is known as Quellung reaction. Clear? Okay. Now, important capsulated organisms. You know, capsules are always polysaccharide. Exception is polypeptide. Okay, bacillus. I already said bacillus, which looks like this. It has one exception. It is the exception because it is made up of peptide and not of the usual polysaccharide. So you know, one polypeptide example is bacillus, and polysaccharide you can say Streptococcus pneumoniae, Streptococcus pyogenes, Neisseria meningitidis, Haemophilus influenzae. So far we have discussed these four, right? And what else? Okay, you can say Klebsiella pneumoniae also. If you can remember Klebsiella pneumoniae. Why pneumoniae? Because, my friend, it goes to the chest. Anything that goes to the brain and lungs are capsulated. Lungs, you can find pneumoniae, pyogenes, Klebsiella pneumoniae, and Haemophilus influenzae. They all go to lungs. And Neisseria meningitidis, it goes to the brain. Right. So they are all capsulated. For now, just remember these 5 examples, and polypeptide example is bacillus anthracis. Okay. If you want to add other examples, you can remember Salmonella typhi and E. coli also. Now what is pili? Pili is hair-like structure. So hair-like filaments. There are 2 types. Ordinary pili, okay, which is of 6 types again, and sex pili. Sex pili is for conjugation, right? That is transfer of genetic material. And ordinary pili, what is it for? It is for attachment. And I give you an example, Neisseria gonorrhoeae. Right? Okay. So pileated organisms, example is, firstly you say pathogenic strains of Neisseria gonorrhoeae, because without this pili, Neisseria won't be pathogenic. Only because of this pili, Neisseria gonorrhoeae is pathogenic. So pathogenic strain of Neisseria gonorrhoeae, one example. And all the gram-negative rods, that is Enterobacteriaceae, E. coli, Salmonella, Shigella, Proteus, and Klebsiella, they are pileated. E. coli, Salmonella, Shigella, Proteus, and Klebsiella. Okay, more you'll learn more about it later. For now, remember Neisseria gonorrhoeae and E. coli or Salmonella, Shigella. They are pileated. They help in attachment. Example, Neisseria gonorrhoeae. And sex pilus is for attachment between male, that is donor, and female, that is recipient, during conjugation. That is, if this is male, this is pili, and this is female who is receiving the information. So this is sex conjugation. Okay, sex pilus helps in this, and normally ordinary pilus helps in attachment to the surface. If this is stable, look at it getting attached. Okay. Now flagella. Flagella are long whip-like appendages helping the bacteria to move. So organ helping in moving or propelling. There are 4 types. They are made up of flagellin protein, mind you, and pili is made up of pyline or pyline protein. Okay, flagella, they are made up of several units of flagellin protein. Structure, they can be monotrichous, they can be amphitrichous, they can be lophotrichous, they can be peritrichous. Now, what is monotrichous? Monotrichous means only one flagella. Example, Vibrio cholerae. Okay. Lophotrichous means that lots of flagella at one end. Imagine one helicopter, and the bacteria will be Helicobacter. Helicobacter. Okay. Then peritrichous, all around, like this. You can say E. coli, okay, and Clostridium, or you can even say Clostridium tetani, okay, or Salmonella. Okay, these are the examples. Or even Proteus. Did you hear Proteus somewhere else? Yes, definitely. When I said pleomorphic. Pleomorphic organisms are those which can change the shape. Okay, they do not have a fixed shape. Pleomorphic. Okay. When we were talking about cocci, bacilli, we also mentioned pleomorphic Proteus. Then amphitrichous means having, if this is bacteria, at both the ends there is one flagella each. This is amphitrichous. Example, spirillum. Imagine a snake. This is bacteria, this is snake. Okay, imagine a snake. Spirillum, spiral snake. So spirillum. So here you'll see the examples. Okay. Amphitrichous, Spirillum serpens, which is like snake, and Spirillum minus. And peritrichous will be E. coli, Salmonella, and Proteus. Okay, Proteus is pleomorphic, and it is responsible for, Proteus produces swarming motility. Remember, swarming motility. Okay, Proteus is highly motile. Okay, it has peritrichous flagella. It is very motile. Then lophotrichous. Lophotrichous is tuft of flagella at one end. Remember Helicopter and Helicobacter species. Clear? Then monotrichous would be Vibrio and Campylobacter jejuni. Clear? Flagella is for propelling, that is movement. And because of this movement, E. coli causes UTI. Urinary tract infection is caused by UTI because of this flagella. And some bacteria species like Salmonella are identified in the clinical laboratory by the use of antibodies. This flagella is antigenic, so it can be identified in the lab. Now, pili and flagella, flagella is longer, pili is shorter, pili is hair-like, right? It is fine, more numerous. Flagella is one or two, or numerous, right? But pili are always numerous, and they are fine. They are always straight. Look, it's straight. But when I have to draw flagella, I'll draw like this. So flagella is curved. Flagella is the organ of locomotion. Pili is the organ of adhesion or maleness. Adhesion because ordinary pilus helps in adhering, and maleness because sex pilus helps in donating genetic material. Okay. From donor male, recipient female receives information. Clear? Then flagella pili. Flagella cannot transfer genetic information, but pili, on the other hand, sex pili transfers genetic material. Now, granules can also be present. Volutin granules, you can remember for now, is present in Corynebacterium diphtheriae. Not very important to remember right now. Just remember granules may also be present. It is present in Diphtheria bacilli. Diphtheria bacilli, which causes diphtheria. This volutin granules present in Corynebacterium diphtheria. It is a reserve of energy. Okay. So specialized structures, capsule, glycocalyx, pili, teichoic acid. Specialized structures are done. Plasmids are extra chromosomal double-stranded circular DNA molecules capable of replicating independently of the bacterial chromosome. They can be transmissible, non-transmissible. Transmissible are transferred from cell to cell. Non-transmissible cannot be transferred from cell to cell. Basically, they carry genes for antibiotic resistance, resistance to heavy metals, resistance to UV, and also genes for the synthesis of pili and exotoxins. Clear? Plasmids cannot replicate independently. Now, what are spores? Spores are resistant or dormant structures of the bacteria. Spore-bearing bacteria is Bacillus and Clostridium. Clear? And fungi is like Candida, you can remember for now, or Rhizopus mucor. But for bacteria, it is Bacillus and Clostridium, B, C. Okay? B and B, C. Then classification of spores can be endospores. Bacteria have endospores, which can be central spore. I said already, Bacillus anthracis. So, terminal spore can be Clostridium tetani. And subterminal can also be some species of Clostridia. Exospores found in fungi. Clear? Endospore is for survival. Exospore is for reproduction. Exospore found in fungi. Endospore found in bacteria. Now, what is the spore made up of? Number one, you see this inner membrane. Okay. Then what do you see? This is the core, which is surrounded by cortex. So number one, core, which is surrounded by cortex. This is the cortex. Core is surrounded by cortex. Then there is one thick wall. Okay. There is one spore coat and exosporium. So basically, if this is core, it is surrounded by, this is core, immediately there is wall which is surrounded by cortex. Then there is one coat, and lastly there is exosporium. I'll show it to you again. If this is the core, okay, this is the wall, this is the wall, okay, this structure, this pink structure is the cortex, this black thing is the coat, and lastly you'll find exosporium. So this is core, and this is wall, core, wall, this is cortex, this is again coat, and lastly exosporium. Okay, these are the structures of the spore. The toughest layer, you see this black thing, or the coat is the toughest. Okay, and this cortex, you think this is the thickest. Okay. Now spore can be central, like Bacillus, I showed you already. And Bacillus, mind you, it also has capsule, which is made up of peptide. It is the only exception of capsules, which are normally made up of polysaccharide. Exception is Bacillus anthracis. There can be subterminal spores and terminal spores. Because of terminal spores, they give tennis racket or drumstick appearance. Spores, you can read the characteristics of spores. They are highly resistant to heat, chemicals. They can survive for many years, especially in the soil. Anthrax can survive for hundreds of years. They are formed when nutrients are insufficient, and they germinate back to the bacteria form when nutrients are available. Then why are they so much resistant? Because they do not have water, and they have lots of calcium, which makes it very thick. It has spore wall, cortex, coat, which are highly impervious or impermeable to other things. Okay, they are very impervious coats are there. They do not have water, and they have rich calcium dipicolinate. Okay, because of all these things, and because of low metabolic activity, they are very much resistant to other things, to damage. Okay, medical importance you can read. Spores are not killed by boiling, remember. They are killed by methods like autoclaving. So if any chemical or physical method which kills the vegetative forms is known as disinfection, and all the methods that kill vegetative forms as well as the spore form is known as sterilization. So boiling is not a method of sterilization. It does not kill the spore form. What is an example of sterilization is autoclaving. Okay. Then sporulation is the process of spore formation. So look at it. Firstly, there is the spore wall. Okay. First, axial filaments were formed. Then there was one four spore formation like this. There is one constriction and one four spore formed. Then this four spore was engulfed. There was one bacteria which formed one four spore or some constriction. Now this four spore was engulfed and it was inside the mother cell. And cortex was synthesized, coat was deposited, and after maturation, there is lysis of the mother cell and spores are released. Clear? Firstly, axial filaments were formed. Four spore was formed next. Then four spore was engulfed like this. Then cortex and coat were deposited. Then after the spore became mature, the mother cell got lysed and only spore remained. Clear? So these are the stages. Axial filament, four spore, engulfment, cortex, coat deposition, maturation of spore and lysis of mother cell. They can not spores can be identified by special techniques which we'll read subsequently. And germination is transformation of spore back to the vegetative form. So vegetative form obtained from the spore is known as germination. What are L forms? Cell wall less forms are known as cell wall deficient forms are known as L forms. Okay. L forms, they can be produced in chronic infections or resistant to antibiotic treatment. That is, if we give any wall inhibitor, cell wall inhibitor, then the bacteria becomes clever. It will form structures which do not have cell wall, so that the antibiotic cannot work on the bacteria and the bacteria will survive for years. So they are formed. So cell wall L forms are basically resistant to antibiotic treatment because they do not have cell wall and they can produce chronic infections. Okay. And what is protoplast and spheroplast? In cell wall of gram positive, if we give lysozyme, it forms protoplast, and if in gram negative we give lysozyme, it forms spheroplast. Protoplast cannot be converted to the vegetative form again, but spheroplast can get back into its original structure later. Okay. Protoplast and spheroblast. Protoplast can't multiply and revert to normal bacterial morphology, but spheroblast, when kept in suitable medium, they can revert back to the bacterial form when the cell wall inhibitor like penicillin is removed. Clear? So this is all about item 1. Thank you.